Good afternoon, and welcome to the Department of Medicine's Grand Rounds. Please take a moment to silence your pagers and mobile devices. Next week's Grand Rounds will be presented by Dr. Mark D. Okusa, Professor of Medicine and Chief of Nephrology from the University of Virginia. Today's Grand Rounds will be presented by Dr. Michael L. Terrin. Dr. Terrin, a professor in the Department of Epidemiology and Public Health, obtained his Bachelor of Arts from Brown University. He received his medical degree from the McGill University before moving on to complete his MPH from Johns Hopkins. Dr. Terrin is an internist with subspecialty certification in pulmonary medicine and an MPH in epidemiology. He has directed or been deputy director of coordinating centers for more than 25 studies with emphasis on clinical trials and cohort studies. From 1979 to 1982, he held a cardiovascular epidemiology training grant at the Johns Hopkins University School of Hygiene and Public Health. He led the Data Coordinating Center for the NHLBI-sponsored multi-center study of hydroxyurea in sickle cell anemia and for the NHLBI-sponsored FOCUS clinical trial comparing transfusion strategies. He was responsible for the data coordination of a landmark study in pulmonary embolism and for the portion of the first NHLBI-initiated thrombolytic agent clinical trials, TIMI-1 and TIMI-2, that discovered TPA's dose and patient characteristic dependent risk of cerebral hemorrhage and acute myocardial infarction. He is currently Contact and Data Coordinating Center Principal Investigator for the NIA-sponsored non-invasive treatment of abdominal aortic aneurysm clinical trial, Principal Investigator for the NICHD-sponsored azithromycin to prevent BPD in urea plasma infected preterms, Principal Investigator for the NHLBI Progenitor Cell Biology Consortium, Administrative Coordinating Center, and Principal Investigator for the NHLBI Progenitor Cell Translational Consortium, Administrative Coordinating Center. Dr. Terrin is also co-principal investigator of the Biostatistics and Bioinformatics Core of the University of Maryland, Baltimore, Older Americans Independence Center, the Claude D. Pepper Center. He has served in a mentorship capacity to associate professors and assistant professors who have established research independence. Dr. Terrin is a faculty member in the Department of Epidemiology and Public Health, Division of Gerontology, and on the department's training grant T32 for the Epidemiology of Aging. He has dedicated his career to the design, conduct, and analysis of clinical investigations with an emphasis on coordinating center data functions. Please join me in welcoming Dr. Terrin to the podium.
Let's see, I've got to turn on my mic. And say thank you for that exhaustive introduction. What do we say? I did a great job. I want to talk to you today a little bit differently about clinical trials than you usually hear, because the question is how to get from where you are with an idea to having done some work. And as we can tell, and we'll go over, there's a lot of book learning and talking, but there's also a lot of help that everybody needs if they're ever going to try to make a clinical trial be anything other than something that falls apart in their hands. So let's start out with a definition of clinical trials. I really love this definition because, one, it comes from Kurt Meinert, who was a professor here before he jumped ship just before he wrote this definition and took a job on the east side of town. And number two, because Kurt is one of the first three academic children of the absolutely remarkable University of Maryland, Baltimore professor Christian Klimt, and I was Chris's last academic child. So Kurt, some 30 years ago, proposed a clear definition among the many various and contradictory definitions that were crowding the field of clinical trials, that a clinical trial is a pre-planned, usually controlled, clinical study of the safety, efficacy, or optimum dosage schedule of one or more diagnostic, therapeutic, or prophylactic drugs, devices, or techniques in humans selected according to predetermined criteria of eligibility and observed for predefined evidence of favorable and unfavorable effects. I think what's remarkable about this definition is that back in 1986, all anybody thought was a clinical trial is something where you randomize people to one group or another group and see what happens after you treat or diagnose them. And a lot of people were sort of uneasy about diagnosis. If you look at the NIH definition of a clinical trial, the definition that requires you to fill out all the extra forms like Form E for a clinical trial nowadays, that definition is pretty much the one Kurt articulated in 1986. So with that in mind, I think it's also important to note that this is what you do. As clinicians, you take care of patients. You apply diagnostic tests, or you administer prophylactic drugs or therapeutic drugs or devices or some interventional technique to people. And so that makes this broader definition of a clinical trial important to all of you, because you want to know whether what you're going to do has a favorable or an unfavorable effect. And where does that stand in the list of things that you might do to create new knowledge? Well, it's absolutely perfectly respectable to publish a case report. Dr. Kleinfelter was a practitioner in Baltimore, and he observed an unusual child in his practice, and he published a case report, and that's the root of Klinefelter syndrome. Or a case series, a series of unusual observations. Dr. Herrick observed that the coronary arteries of patients who succumbed to acute myocardial infarction had thromboses in them, and he published the series, and that's the foundation of our modern knowledge of coronary artery disease and myocardial infarction. There are cross-sectional studies, case control studies, historical cohort studies, prospective cohort studies. Clinical trials largely sit at the very end of all this work and need those other earlier efforts to justify them as preliminary data. You're going to have a great deal of difficulty securing support to conduct a clinical trial from somebody outside of this institution unless you've got strong preliminary data to support it. And the good news is that our ICTR is about a lot more than just clinical trials. It's the place you want to go if you need help to put together the basic preliminary data and infrastructure to get your clinical trial going. So here's the clinical trial paradigm. It's what you do in a classroom to get a clinical trial going. Basically, you need a target population. You need an idea what it is that you're going to do for some good for the target population, a screened population, that portion of the target population that you can get your hands on, and then when you've applied your eligibility criteria and asked for consent of that screened population, your study population. Clinical trials do not have to be randomized. This is the chart of the simplest kind of randomized clinical trial, but instead of randomization, you could have your groups separated by time into before and after treatment, or you could have your groups separated by an instrument, some natural occurrence or progression of approaches to healthcare in different settings that would separate the groups who can then be the recipients of the treatment or diagnostic procedure that you have in mind, and then followed for outcomes, and then with those outcomes ascertained, proceed to the comparison of those outcomes in a data analysis. Now, this is what you get in textbook learning, and you could have fun fleshing some of this out as more textbook learning in the course I teach, Prev 803, Clinical Trials and Experimental Epidemiology. But if you want to get a clinical trial or the preliminary work for a clinical trial done, you got to start sweating the details, like how are you going to find the people who fit the Aristotelian category of your population? Or how are you going to get the treatment or the diagnostic test that you're going to give them and maintain it and have it available to dish out when you get your hands on an agreeable and eligible individual? And then how are you going to make sure that that treatment takes? And then how are you going to make sure that you don't lose track of that individual so that you can find out what happened to that individual and then compare the results either in the change of that individual's status or between two groups? And how you do that involves a tremendous series of nuts and bolts. I may make reference to some of the specifics of the nuts and bolts as I look around the audience here and see some of you who I know have struggled with this problem. And that's not textbook learning, and it's not something you can do for yourself, especially if you're early in your career. It's something that you need a lot of help for. One of my favorite authors in moral philosophy, C.S. Lewis, who most of you know perhaps because of his Narnia books, wrote, "I can do it myself is one of the surest ways to end up in a very undesirable place." We are very, very fortunate here at the University of Maryland, Baltimore, now to have on our campus an Institute for Clinical and Translational Research. This is a collaborative institute with Johns Hopkins and other partners in the city of Baltimore, I guess outside of Baltimore in the healthcare practice with which we're affiliated. But the main axis of this runs from east to west Baltimore between Hopkins and the University of Maryland, Baltimore, and has a tremendous amount to do with making clinical trials feasible. And from the layout of the ICTR here, there are remarkable resources available that everybody's going to need for a clinical trial. Just starting at 1 o'clock, the biostatistics resources. You're not going to get a clinical trial going or prepare preliminary data for a clinical trial without an analysis. You're going to use that preliminary data for a power calculation. Biostatistics, if you're going into a randomized clinical trial, establishment of a randomization system. That sort of nuts and bolts is somebody you need to sit behind a computer and generate a randomization schedule for you. And the good news is that help is available through the ICTR if you put in an application on the ICTR website. Similarly, you may need stakeholder buy-in depending upon whether you have immediate access to patients through your own practice or services, or whether you have to go out into the larger community to find the patients who are going to be your partners in your clinical trial. And if you have to go outside, once again, our ICTR is prepared with cooperation from Dan Mullins, who's just a whiz at this in the School of Pharmacy, to help you get in touch with the stakeholders and get your work driven by the ideas that are perceived in the community to be of value to the community and open the doors to access to your patient population. Informatics for handling of the data, a voucher program that will support many of these functions that you need upon an application, an ATIP grant program, which may be small in number but is quite mighty in effect if you can get those resources through competition within this campus to support the conduct of your clinical trial. And the many, many CTSA cores, which can be grouped as research and methods cores, advising you on how to design the trial, a network capacity core that we're going to discuss more in the form of trial innovation centers and recruitment innovation centers, drug discovery and development core. Let me say that in addition to the national CTSA cores, where those resources are available for pharmaceutical manufacturing and development of formulations and drug design, we have an incredibly powerful school of pharmacy, and that pharmacy is part of our CTSA also. I've found the Dean, Natalie Eddington, to be a wonderful collaborator, and her faculty have helped me and Rose Viscardi out in our clinical trial of azithromycin in very premature neonates. And again, she's helped me out again in the non-invasive treatment of abdominal aortic aneurysms to figure out what our patients' doxycycline levels are. So the resources are there. If you're worried, for example, about is this pharmaceutical that you're giving patients really getting to the levels that you need in the blood, there is, through our CTSA ICTR, the capability to do that. And that's something that you don't get just by dreaming it up theoretically in Michael's class. So what are some of our ICTR resources? Well, there's a navigator. If you want to see what the resources available here on the UMB campus are, I recommend highly that you go to the ICTR navigator at umaryland.edu. One of the most valuable things to start up if you're at the stage of an idea formation is a studio, or early stages in thinking through your project. Those are available right here on the campus by going to one of our charming navigators on the ICTR navigator, putting in an application, and assembling a body of interdisciplinary experts who will, free of charge, advise you on the project and come to you where you are, whatever the nitty-gritty specifics you need to talk about to bring your project ready to its preliminary data collection stage or to its grant application submission. That's what the studios are here for. And then there's Research Match, which is a national registry of volunteers, people who are interested in joining research, 144,176 at last count. There's a fairly substantial number right here within 50 miles of the University of Maryland, Baltimore. These are people you can draw upon who may or may not be exactly what you need. Because these are people who are standing and waiting, they largely represent relatively healthy individuals or chronic conditions. If you're looking for acute presentations, you've still got to work within whatever your fishnet can catch for you here. With that, just only pause and say I'm delighted to take questions throughout this presentation. So if any of you have questions, please ask them. Anything I can't answer, I am quite sure our ICTR leaders seated 3 in a row in front of me would be delighted to talk to you about. But that sort of nuts and bolts that you need is what you should be considering whatever level you're at, whether you're a resident seeing something interesting going on among the patients who you see on the medicine service, or a junior faculty member or a senior faculty member. So right here at the University of Maryland, Baltimore, we have the ICTR, and we offer the studios, as I discussed, and Research Match is available to all of you through the ICTR who are looking for local or even more remote patient access. Biostatistics I described, and the informatics can provide help with data management, and there are programs of awards and vouchers through the ICTR to mobilize those resources as well as the other resources I mentioned. Community engagement, which if you're going to have to put in some clinical trial grant applications, you've just got to document you've got. The other resources, almost too numerous to count here right now, can be found if you follow the take-home message, navigate the ICTR website, go to it. There are then some more resources that are available through the University of Maryland, Baltimore, Johns Hopkins University Axis. Here, Dan Mullins and I, Steve Davis, a couple of others represent the University of Maryland interest in this. At Johns Hopkins University, the interest in the clinical trial or clinical investigation end of this axis is represented by Dan Hanley, who's very familiar with working with us. He made tremendous contributions to our Older Americans in Independence Center in his capacity as a neurologist and a collaborative investigator. Charlie Flexner and the staff who work with those individuals at the Johns Hopkins end of this at the corner of Pratt and President Street, notably Cassie Land Lewis, for example. So they have within their grasp a Trials Innovation Center, a Trials Innovation Network, a Trials Innovation Unit, and a Recruitment Innovation Unit that are local to us here in Baltimore, and I'll come back in a minute or two to discuss those. The take-home message is draw upon those collaborative resources to enhance whatever you have to support you here locally. And then there's the national research resources, and those include the National Trial Innovation Network, the Johns Hopkins Trial Innovation Network, Trial Innovation Center is one of 3 trial innovation networks, and a National Recruitment Innovation Center, as well as Research Match, which is a project of the national CTSA effort. So in addition to visiting our ICTR website, I would advise you to visit the CTSA national website to see what the resources are there. Now, what does a Trial Innovation Network do? Well, there are 3 of them, and their idea is to support multi-center clinical trials and studies. But the emphasis is on multi-center, and to help them get to single IRBs, to use the formats that they've developed for master contracting that the clinical sites and many cores have agreed to. The idea is to speed along clinical trials, translational research, clinical translation and science discovery, and to help those studies have quality in them by design, and to advance their recruitment and patient engagement. Now, why are these the focus of the Trial Innovation Network? Well, this is mostly of concern to the more senior individuals in the audience. The more junior individuals are probably thinking of their own personal projects or single center local projects. And the Trial Innovation Network grows out of the sense that, and this has happened to me, you apply for what the NIH considers a long project, 5 years, and it ends up taking you 7, 8, 9 years. The idea here is to speed things along so that clinical trials proceed more rapidly to come to their conclusions, so that the information gets into the public domain and subsequent clinical research can build on it. So this is, again, for multi-center studies. And getting a single IRB is no mean feat. Most IRBs in the United States do not want to take on single IRB responsibility. And the reason is it's a tremendous amount of work. You've got to credential over many, many different clinical sites. Most of the clinical site IRBs are not well enough supported to take on some of these massive tasks. I've been extremely grateful to campus here, which has been willing to take on single IRB responsibilities for 2 projects that I've proposed. But let me let you know, if you need a single IRB, you may well have to go to a commercial IRB, or you may have to go to a site that has an IRB within your consortium that's willing to take on single IRB responsibilities for what might be as many as 50 sites or more. Our IRB is very generous with its resources, and I think absolutely invaluable. I have almost no patience from my colleagues who view the IRB as an obstacle to be overcome. From my point of view, the IRB represents individuals who have in the past kept me from doing things that I would regret for the rest of my life. And I don't always agree with the IRB, but I do believe in the rule of law, and I believe in all the help that I get. So the Trial Innovation Network then can help you set up a single IRB if you have a multi-site study that is not within the capacity of our IRB on the UMB campus to handle. I've discussed the master contracts. Again, contracting can take just months and months and years unless you have something that everybody has pretty much agreed to in advance. And designing for quality in advance so that you have adequate monitoring and resources dedicated to assuring the quality of your study is well worth it. And then the one thing that slows up clinical trials more than anything else is recruitment and engagement. And I always think of my friend Barry Davis at the University of Texas, Houston, who led the Data Coordinating Center for a clinical trial of antihypertensive agents, all-hat, that was originally planned to have 50,000 patients. And I once bumped into him partway through the study and asked him how things are going. And with his usual laconic response, he said, well, depends. We've got 30,000 patients. And I said, that's fantastic. And he said, yeah, but we're 10,000 behind schedule. So the resources that the CTSA can offer on top of Research Match here to help you rationalize your recruitment are enormous, and that's stepping back a little bit from this multi-center clinical trials challenge, because in the JHU UMB axis there are considerable resources if you go work with them in advance to assure that you've got patient engagement and recruitment settled. So the Trial Innovation Center that has one foot here in Baltimore is the one that runs between Tufts in Boston and Hopkins. The Trial Innovation Center here has as its goal, largely related to the Hopkins brain injury outcomes studies commitment, to develop new methods for multi-center clinical trials, especially those that are going to be applicable to brain injuries, to aging, to Alzheimer's disease. So if you want extra help from the Hopkins group, their TIC is a place to go for this. They are highly selective. They support studies. They are part of the network, the other TICs across the nation in the Trial Innovation Network, and the Trial Innovation Network being composed of the TICs and the research centers and the national CTSA resources. So if your proposal, and again, this is for multi-center clinical trials, so this is pitched mostly to the more senior investigators here, is in development and needs extra help and maybe some resources, you can go to the TIC to ask for their assistance. Some of the things the TIC at Hopkins offers are free of charge. Some of the things the TIC offers are fee-based, so you've got to find a resource that you can use to pay for what you're getting from Hopkins. That's especially with regard to data management, for example, or to this provision of specifics for recruitment for the clinical trial. And then behind all this is the Trials Innovation Unit. Now, the Trials Innovation Unit, again, on the UMB Hopkins axis, located within the Hopkins end at Pratt and President Street, is more interested in single-site trials, but it's interested with those single-site trials that have potential for multi-site expansion. So if you have a single-site trial that might, say, be a phase 2B study, and you're planning to go to many sites, if your phase 2B study turns out to be promising for your intervention, it would be good to approach the Trial Innovation Unit and start discussions early. They are also prepared to help you with multi-center trials in preparation for TIN submission, so that if you've got a single site trial and you're planning to go to a multi-site expansion, and you're going to want the trials intervention network to accept your application, to provide resources that will perhaps make you a competitive clinical trial for funding from one of the National Institutes of Health, or you've gone to the Trials Innovation Network already with your trial, and they say you're not quite ready for prime time with us. They'll refer you to resources for an undeveloped protocol to prepare it to be ready for the TIN. Or if the TIN looks at a clinical trial and says, yeah, this is pretty good, we want to go with it, but we want the Trials Innovation Unit to work on it, they'll assign it to one of the Trials Innovation Units. One of the most interesting things that's available here, in addition to the studios here at UMB and Research Match and the advice that you can get from the ICTR folk here on recruitment, is that our other end of the axis at Hopkins operates a recruitment innovation unit. This is run by Cheryl Himmelfarb, and I think you can get access to this by application through Cassie Land Lewis. And they offer consultation services as well as referrals to bodies that are going to help you out. So if you need help with community engagement, which I think you could probably get on this campus by going through Dan Mullins, you could go to the Recruitment Innovation Unit at Pratt and Pratt and President Street and find other community engagement access. If you need assistance with ethics or regulatory, you could get a consultation with that group too. Again, I think your best stop would be locally because of the strength of our IRB and our ethical and regulatory advisors here through our own ICTR, but that resource is available. They also have a resource which you'd have to pay for, but it's marketing. They have a formal marketing structure in which they'll go out to the community and market your clinical trial to the community. They'll do that for academic studies, and they'll do that for commercial studies for a fee. but I think marketing can on occasion be quite valuable. I happen to be personally much more of a believer in shoe leather. I like to see University of Maryland, Baltimore investigators and their staff go out and press the flesh and find patients for their studies. One of the things that frustrates me in some clinical trials is when I see the investigators or the staff going through the motions. Yeah, we're mailing out flyers. Yeah, we're setting up a table at the market and putting flyers there, but there's nobody at the table. I think there's just absolutely nothing like individual contact. But if you need marketing, there is a formal marketing organization there at the foot of the Jones Falls Expressway, and you can have access to it. They also help with recruitment, and their help with recruitment, I think, is beneficial. It's of a slightly different genera than Research Match or but different from the help you can get from the ICTR here. Their help with recruitment is in the nature of knowing local practices and researchers, ranging from pediatrics to geriatrics, with all the surgical specialties and medical specialties in between, and covering dentistry. And so they know the investigator-friendly or the research-friendly practices in our immediate area, and they will put you in touch with those investigators to help you get patients. Likewise, they can provide coordinators. That too is on a fee basis. And then moving on from the Baltimore area, the Recruitment Innovation Center is the one I found is located at Vanderbilt. It's part of the national CTSA effort. And I think it's a remarkable forum. If you're interested in recruitment, I think you should go to that website. They have a newsletter. You can read the newsletter. It's full of discussions and tips on do's and don'ts for recruiting patients, how to access special populations, and if you make use of the CTSA national research structure, you can get access to their consultation and assistance with designing your recruitment. They aren't on the boots on the ground like Research Match. Here's a list of patients, see whom you can get from it. They're not boots on the ground like the RIU at our collaborative ICTR. Here are the practices in the area where you might be able to find the patients. The practices are sympathetic. They're not boots on the ground like here, where you and your colleagues should be able to go to press the flesh on this campus, but they are very helpful. And so what happens at the end of a clinical trial? Well, just before moving back from the world of the practical, what you've got to do to get help from the School of Pharmacy or from our ICTR with recruitment and navigating it. You can also be relatively informal. Anybody here who wants to can come to my office, and you can get all the advice free of charge that you want from me without getting a studio, or even more advice with the studio. I assure you that advice free of charge is worth what you pay for it, but it's there for you. When you get to the end, then you're going to have a clinical trial, and it's back in your hands. You can get the experienced people around here to give you advice, and I can't recommend that too highly, because the biostatisticians will analyze the data, but at the end of it, you've got to make sense of whether you have favorable or unfavorable results. And what you've got to do then is at the start, at the very beginning, the challenge in the design is to solve, possibly going through this over and over and over again. Oops. the problem of matching the patient population or disease condition with the treatment and outcomes planned so that the final data analysis answers your question, which you've got to frame as a testable null hypothesis. I'm going to reject this idea as null, or I'm going to fail to reject the idea as null with some conviction. And so all along the way, you have a logical challenge that's based in, have you got the nuts and bolts working, not just the theoretical diagram of your study, but if your study finds a difference, you've got to go back and check every spot along the way that the difference wasn't due to the introduction of some bias. Was your placebo, for example, not really a placebo, but in some way harmful? And we've had a tremendous amount of feedback from the FDA of late with regard to proposing excipients as placebos in inhalation or injection drug clinical trials, with the FDA saying, how do we know what you're giving us in placebo isn't harmful, and what you're showing us with your active agent isn't just that your active agent is less harmful than the placebo itself. And you've got then the difficult choice between, should you have a usual care clinical trial or should you have a placebo clinical trial? And that then ramifies into many other choices. In a placebo-controlled clinical trial, it's a lot easier to retain your patients than it is in a usual care clinical trial where a patient may have given consent in the hopes of getting the new experimental treatment, and then upon finding out that he or she isn't getting the new experimental treatment is less bonded to you, less interested in the clinical trial, and harder to follow up, even if you tell that patient the truth, which is in the large majority of clinical trials, the patients who get the placebos are the lucky ones. Right? I mean, only half the clinical trials that get proposed to the FDA get labeling, and a lot of the development falls by the wayside before they get to FDA applications. So you have to check to find that your method of ascertainment of the outcome didn't introduce bias. So you've got to have that solid catchment to get the outcome in all or just about all of your patients, and without interference from knowledge of the treatment, which might make the interpretation of the outcome differential across the patients. Or if it's not a randomized trial, knowledge of whether the evaluation was done before or after. And if any of you think this is trivial, let me tell you that you should only go to radiology and talk to them about getting CT scans, where the earlier CT scan is not distinguishable with regard to its time of performance from the later CT scan. Those are the sorts of nuts and bolts you need extra help to conduct. Similarly, in studies that find no difference, you've got to go all up and down the clinical trial structure, the nuts and bolts, to make sure that your clinical trial was sensitive in establishing the differences. Did you save some of the study treatment if it's a pharmaceutical? Did you assay it to make sure that the pharmaceutical active was in the treatment kits labeled active and the pharmaceutical placebo was in the treatment kits labeled placebo? There have been studies where they've been mixed up. Other nuts and bolts, did you, as I say, check the circulating levels of the study treatment that you're interested in to make sure that some people weren't in the control group getting the treatment on the side if it's a pharmaceutical, and other people not getting the treatment, testing your bioavailability or your fidelity or an objective measure of your adherence. And once again, I want to say that part of our campus organization, the School of Pharmacy, has been just invaluable in that. So to finish, What are the rules of the road for any of you who want to do clinical trials, whether they're single-center clinical trials to be performed here or multi-center across the nation or internationally? Well, they're the Navy priorities. First of all, you've got to float. That's your infrastructure, your nuts and bolts. That's everything your ICTR can help you with. And once you're floating, then you can move. Because you can't fight if you can't float or move. And moving is recruiting those patients to get them into the studies, and then you can go ahead and fight. You can treat the patients and you can evaluate them. But you aren't going to get to your third priority if you don't have all the nuts and bolts, if you aren't recruiting. And if you think this is easy, well, going back to Shakespeare about kingship, any clinical trial principal investigator who sleeps well at night lives in a fool's paradise, because uneasy lies the head that wears a crown. That's it. Thank you.
Thank you so much, Dr. Terrin, and thank you for stepping in at the 11th hour. Superb. Questions, comments? Stella?
That's something that is actually attempted, and sometimes it's successful, and sometimes it's not. And I think what it comes down to is the devil is in the details. Yunan Shan, who is part of our ICTR, I believe, in the School of Nursing, actually has had a web-based clinical trial some years ago, and it was a spectacular success. It was something that depended upon transmitting the intervention through the web, so everything was in one package. It's a bit more difficult, but possible, to recruit nationally and ship drug. Now, I think you've got to come back to our ICTR and get a whole bunch of regulatory help, because there are restrictions on what you can ship via postal or FedEx or UPS service, and there are state-by-state restrictions on distribution of pharmaceuticals. But this can be done, and then there's behavioral interventions, and you can recruit through the web. You don't get a nationally representative sample in recruiting, but you can recruit through the web, and you can institute behavioral interventions, whether they're yoga practices or smoking cessation practices over the web. And I think those have tremendous room for success. And you can do really creative things, because web doesn't have to be broadcast. It can be receive data from your patients and feedback and help with recruitment and inquiries and performing the intervention as well. A tremendous advantage of this is that you can structure your internet connections so that your patients don't all know each other and don't contaminate each other, and all the patients in the same waiting room is one of the great bugaboos of the old-fashioned body is there clinical trials. You can do some creative things, and you aren't the only person in the world who's thinking about this. I think the major frustration with this use of electronic media in clinical trials is that the status of electronic health records, which you would think would be a goldmine of information, is just a nightmare across the country. Nobody's interoperable with anybody else. I think one of the things I'm most proud of in our ICTR is the commitment to make our records and Hopkins records interoperable. There's been some success with this. On a state basis, Rose Viscardi in our azithromycin study has been able to track down very premature neonates 1 and 2 years with the assistance of health records to which she has access through our Baltimore systems, because these kids move from different households to different households and nobody's refusing contact, but they can be they're there, but they can be hard to find, and I'd love to see more progress.
You referring to CRISP?
CRISP is not the only one. It's also the Hopkins and the Maryland system, which may come closer to each other.
Right. So yeah, watch this space. So we have some T32, which, as you know, are training grants with John Pierre Ralphman, Simeon Taylor, Andy ran one. Do you have concerns about the pipeline of junior faculty, postdocs going into clinical research? Any ideas in the last 3 minutes of how you solve that, untie that Gordian knot? Because we have concerns as well.
I think that it's hard for a clinical trainee to feel comfortable that there's a sustainable career path, given the challenges of NIH funding, and it's also challenging, I believe, for institutions to find internal money to support people to the point where they get external funding. And I basically think it's a very attractive career if you can do it and if you can be successful. But I could be wrong that I think many people sort of are just concerned with the practical things you can do about the sustainability. You would know more about that than I, but that's
Maybe I think it's a good idea. I appreciate the suggestion that the Jefferson suggest that the Roy and we think about the fearful of being resources to the life. Because it's a very low cost thing that they benefit.
So I just want to very quickly follow up on something that Jean-Pierre said. So thank you, Mike. There's a lot of nice things about the ICTR. And so the ICTR, CTSA have brought in 5 new training programs, both at faculty level and at pre-doc and post-doc. The post-doc was particularly challenging because the amount of individuals applying for postdoc fellowships has dropped 10% over the last 4 years. We filled, Hopkins did not. And we filled because of outstanding staff and faculty, so sitting in the front row, and also GPILs. So we all came together, the 7 schools came together out of the graduate school, and we succeeded. So I think it can be done. But as Jean-Pierre said, it's step by step, and you've got to learn to float first. So. Kathy, do you want to try? Okay. Do you have a quick question or statement? Yes. Yes, yes, absolutely. Okay, thank you again, Michael.
That was a fiery finish, Steve.
It is.
Attention.